in this panel session from the different organizations or countries. We have brought some key people in this session, and idea is to invite them, to request them to share their vision, what should happen in their respective countries or in their organizations, or with the overall objective of enhancing crop productivity, profitability, nutrition, etc. So these are some of these ideas, and I think that we are having the top people in this session. Because we have limited time, we will also like to see the interaction of the audience with the panel session specialist. So we will be doing, or we will be having short or brief interventions from each of the speakers. But before that, I move ahead for introducing our speakers. Their brief bio is already available in the abstract book. So in this context, I would like to invite first Professor R.B. Singh, who is the Chancellor at Central Agriculture University in Imphal, India. And Professor Singh has served Indian agriculture as well as international agriculture in different capacities. Again, will not go in the detail about his bio because he is a well-known person, and secondly, this is already available in the book, but would like to mention one quick thing, that he has been awarded the Padam Bhushan by the Honorable President of India, which is the India's highest civilian honors that can anybody get. So we are very pleased to have you, sir. So please join me in welcoming Professor R.B. Singh as a co-chair here. Now, I would also like to invite Professor Eric Danka. He is the Director for West Africa Center for Crop Improvement, and he is based in the University of Ghana. He has again served several important organizations and also several advisory committees of the CGIR and CRPs, etc. So we are very pleased to have Eric Danka for this co-chairing this session. And now I would like to also invite other panel session specialists. So from India, we have big organization, Indian Council of Agriculture Research, and we have taken one director as a representative of ICR, and we have Dr. N.P. Singh. He is the director of Indian Institute of Pulses Research, and he plays a very key role in ensuring the pulses research and development in the country, but here he will be representing ICR. So please join me welcoming Dr. N.P. Singh. Now we have Dr. Liao Baosu, and he is representing here China. So he is the Director General of Oil Crop Research Institute for Chinese Academy of Agricultural Sciences. ICRISAT is having long-term collaboration with Professor Baosu, and we are very pleased to have him here again. He has won several awards, served several important positions in China and internationally. So please join me in welcoming Dr. Liao Baosu. Now I would like to invite Dr. Paul Kimirto, and he is the director right now for the Agro Science Park and at Egerton University in Kenya. Again, he has been long-term collaborator of ICRISAT, not only ICRISAT but CIMMYT and many other CGR institute. He plays a very important role of using the modern technology in crop improvement, not only in Kenya but in general in sub-Saharan Africa. So please join me in welcoming Paul for this session. And then would like to invite Dr. Khalid Amri, and he is the director of Khalifa Center for Genetic Engineering and Biotechnology at University of Emirates, UAE, and I have visited that center. This is fantastic. So Khalid, we should have, okay, yeah, so please, yeah, sorry about that. And Khalid is fantastic scientist, of molecular biologist, biotechnologist. So please, Khalid is here. And then I would like to invite Dr. Chike Ma, and he is representing FAO. Right now, he is leading a team of the seeds and plant genetic resources of the plant production and plant protection division of FAO. He has served several organizations, including CEAT, IAEA, and you will find his name in many other panels or so. So we are very pleased to have you, Chike, here. And after that, we would like to invite Dr. Victor Navasu, and he is the Senior Fellow and Senior Program Officer for Peanut Sciences for Mars Wrigley Confectionery segment of Mars Incorporated, and he is representing the private sector. So, and again, he has played very important role in the genome sequences of peanut, and also his companies for cacao and for many other countries, playing very important role in sub-Saharan Africa and Western Central Africa. And now I would like to invite Dr. Oscar Raju, and he was already introduced on the first day. So earlier he was at ICRISAT, but even he served several other organizations. And right now he is the lead for that molecular marker team or genomics assisted breeding related thing, technology lead at Dow. Well, they changed the name here, Dow DuPont, based in College Station. So please join me in welcoming Oscar here. And Oscar is representing big company here. And now I would like to invite Mike Butterfield. He is coming from the CTC company. He is the manager of the sugarcane breeding, and CTC is one of the largest private sugarcane research based in Brazil. So basically we are having the representation from South America as well. So Mike, you should be here. So please join me in welcoming Mike. Unfortunately, you need to go all the way around from there. Sorry about that. And we also have Dr. Arvind Padhe. So he will be probably joining a few minutes later. And he is our country relations officer for ICRISAT based in Delhi. He is the IAS officer and also holding the doctorate degree. He is having good experience of agriculture development. So he will be joining us later. With these remarks, now I hand over the session to Professor R.V. Singh and Professor Danka. So please, sir. Yeah. And please join me in welcoming all those panelists once again here. We have the mic if you would like to come here. Otherwise, you can speak from there as well. So, yeah. Okay. Otherwise, all panelist speakers, they can speak from there. So we are having the mic. This will be easy to rotate. Thanks.
Yeah, thank you. Very good afternoon to each one of you, ladies and gentlemen, distinguished participants, Professor Paroda. I must say that this panel is something which is absolutely different from other panels that we had. Raju, thanks for the opportunity you have provided us. I would also like to take this opportunity to thank David and the entire ICRISAT system for this outstanding achievement that you have had for the last 3 days in terms of various presentations. I think it's most timely conference where science, technology-led transformation of production systems, the entire national, international systems can really see to it in a collective way. As a matter of fact, I would like to very much congratulate the wholesome approach which has been taken, and this panel is a reflection of that total wholesome approach. The 10 persons who are present here, including me and my distinguished co-chair there, represents the entire world in a way. All the continents are here. It also represents the national and international. We have the FAO right in the middle, as you can see there. And then we have the Latin America, Caribbean representation, Africa, Asia, you name it. China being here, this again makes the difference. India and China together, you can see what proportion of the world population and so on. And of course, the outstanding colleague from US and so on. So friends, I thought this is a wonderful panel to be with, and this I think is absolutely important. Another thing which I think is important for this panel is that not only the public, private, international systems, all possible stakeholders in one way or the other are represented. It's not only the public, but also the private sector is very comprehensively represented here. So I thought from that point of view, the perspective that we are looking for, the application perspective of the genomics that we are discussing today puts us right in focus in the contextuality where we are. And this contextuality must be seen in the backdrop of the Sustainable Development Goals, and the first 3 goals, that is the alleviation of hunger, undernutrition, poverty, and of course the resilience to climate change and the environmental protection. Also we see we are today on the same page as the whole world, that the zero hunger, the ICN2, the nutrition being at the center stage of the world development, The International Year of Pulses we had, and this is where I think could not think of a better occasion than this occasion to remind you that what we discussed at the International Year of Pulses and here is absolutely in line, and Rajiv again for this opportunity, and David again for this opportunity to remember the 2016 with the culmination of 17 coming up. Here, I think the pulse is being put in the center stage for nutrition, and genomics for nutrition, hunger alleviation is something very outstanding message which we will be able to put through. Particularly coming from countries like India, the governments have been also looking at us with a very, very focused eyes, and I would like to remind you that today, the Prime Minister's call in India, doubling farmers' income to transform India to a new India. I think these are the ways that we have to look at it together. And I find with all these promises, I find that we have still a long way to go in meeting our commitment. FAO would be able to tell it more comprehensively, our commitment to alleviate hunger, poverty, and undernutrition. And this, I think, should be our guiding principle to see that how we can apply the genomics, which in the last 2.5 days, the wonderful presentations, I think the very rich, absolutely scientifically rich contributions which have come, have really made a platform from where now we must be able to connect the policies. These wonderful discoveries, wonderful leaderships in sciences do not ultimately lead you to the ultimate fruits that you have to deliver, and therefore the intervention or the trinity of functions of science, policies, and implementation must be combined, and this is where I thought the application strategy has to be talked about. Then we must keep this in mind, that it's not only science per se, but it's the science with a human face. And this also happens to be the logo of ICRISAT, and it is this thing that will be important. Also, the science will not deliver in a vacuum. And so, if science with discovery, then science for delivery. If this is also ICRISAT slogan, then delivery would require that there is a way to walk, and you should be able to walk only when there is a proper policy and pathway. And hopefully, a pathway will be decided when we go out of this room this evening, or in time to come, within a week or 10 days, this will be decided. I would finally say just one word, that while the congruence of the various scientific discoveries, the wonderful linkages that we were able to see and read here can only play an important role if we are able to convert it into something which you can hold it in your hand in a tangible manner. And while we do that, we will be doing that, I have a few questions which possibly would probably engage you for some time to come, and we must be able to answer those questions and probably take note of that. The frequently asked questions that would be that what is happening, we have been always talking about for the last several years, we are at the same place running rather than not moving forward. If we look at the rice hybrids, if we look at the other hybrids, including the hybrids in the legumes, pigeon pea also. So we must be able to see what is happening to the hybrid technology as such in a large number of crops and commodities. We have been talking about it, but we have not moved very far in this particular pursuit. The state of transgenics, of course, genomics is not exactly what is referred to, but then transgenics in any of these crops, all these crops, a lot of publications, a lot of good work being done here and there, but we are still nowhere nearer that we can say we are close to use it, including the master transgenics, and we have to fight the rules, regulations, many of the baseless fears, many of the emotions which come in the way of science, and possibly this is a group like this who can enlighten, can send the message that probably science should not be denied to serve who need it the most. and this is being done, and we should be able to be very frank and bold about making some of those programs. Third, we notice here that over the years, what used to happen with the CEG centers, a lot of pre-building material will flow, lot of interactions with the national programs will be there. We heard a presentation, thanks Norwegian, German support, that many of the programs are running in the pre-building programs. But I find the nations must arise to the occasion. The pre-building program is still very weak, and to have pre-building, you have to have outstanding scientists, and there is something going wrong somewhere that we are finding this is not taking up. I would urge very strongly what can be done to really connect the nations with internation, and nation and local and global is no more differentiated. The high interdependence of local and global is widely known. Local, actually, if you see, is the way forward. This is not happening. And if this has to happen, this must be aligned to the process. Finally, of course, we heard Mike Bevan. I was able to talk to him and say that, look, you're talking of can the epigenetic manifestations be silenced? In the plants, if they can be silenced, they are being silenced in the plants. Can we think of the generations of people who are hungry, who have remained undernourished, nurtured? They are deprived for years together. Is that getting epigenetically fixed in the system? Do we have the intergenerational transmission of poverty and hunger? If epigenetic silencing is taking place in crops, can there be epigenetic silencing of this kind of hunger-led deprivation of the poor and hungry? I leave this question to you. You are all great scientists. Please think of it. We heard that now you can really interact with the plant genomes, with the human genomes, with the animal genomes. You can use the various technology of one with the other to develop absolutely modern, focused, precise plant breeding. But I think there is this, we live in the fast-changing, interconnecting world. The message is connect and share and grow, which is not happening. I will urge that this panel, because you represent the various parts of the world, you represent the public-private partnerships and so on, you represent the academia and those who are involved in development, you are all involved in the process. I urge, please look at it and give us the guidance. Thank you very much for this opportunity.
Let's give him a round of applause. Thank you. Yeah, okay. I was wondering whether Rajiv thought about the 2.5 hours that he gave us, but I believe he knew the chairperson he chose. Thank you very much, my more distinguished chairperson, for the introductory remarks. We have to keep you awake for about 2.5 hours. I believe we will. We have a very distinguished panel over here. I just would like to thank Rajiv for this honor done me, invited me to this very important anniversary celebration. Rajiv, thank you very much. I believe that it's because we go back when your center was 1.5 years old. Thank you very much for the conversations that we've had over the years. I wouldn't speak for long because my distinguished co-chair has spoken. I would like to first invite the first speaker to speak to the topic, Genomics Applications Perspective, looking at what has been done at your institution and the future of genomics, present and future. And perhaps if you have any ideas that you'd like to share with us, we would be most grateful. So, Dr. Singh, would you kindly speak from here? It's preferred that you speak from here.
And good afternoon to all of you. Very eminent panelists, chaired by Professor R.B. Singh and Dr. Eric, colleagues from different parts of the world. Good afternoon once again. Friends, you know that India in the previous year achieved the record production of food grains, including pulses, where we had always a big deficit. We were, deficit by 4 to 5 million tons. But we had a record production. It was all backed up by a strong government policy. So the government of India is all determined to push up pulse productions. Our in field of genomics, lot of developments has taken place in the country. Now we have a mass produced rice, wheat, pearl millet, and again, in the maize. But the pulses, which were not so much benefited with the tools of biotechnology, in recent years, tremendous progress has been done. Dr. Rajiv Vasni, along with national program, you all know that we have a first chickpea genome draft sequence, then the PGNP, and that is further improved over the period of time. Now we are in the stage to use those information already generated in the breeding program. And I'm delighted to tell that in chickpea, at least we have a couple of products that has been already under testing. Maybe in a couple of years, they will be able to see the light of the day. And particularly for wilt resistance, we have tried to pyramid genes for race 1, 2, and 4. And again, drought and heat tolerance gene has been pyramided. This is the one. And secondly, in PGNP also, very happy situation. We are trying to combine SMD resistance and again to resistance to fusarium wilt. Very similar situation is there in the crop like mung bean, where again genome sequence is known. We are making use of those genome sequence, and we are in the process to develop a product. Maybe in another couple of years, it will also enter into All India coordinated trials. Our two teachers are here who 2.5 years back initiated program in transgenics and genomics. Transgenic, however, one side genomics moved very fast and we are in the product mode now. Transgenic, bit we lagged in the legume crops because, in the beginning there was a kind of information that was generated. I should say there was not much effort, so they were called to be recalcitrant. Now, over a period of time, the, regeneration transformation protocol was put, now there are throughput, I should say, protocol. And at least there are two places, the one that Assam Agriculture University with different collaborators, the Helicoverpa resistance BT transgenic now in the trial. Similar way from our institute, we have been permitted by RCGM, GEAC, two different regulatory bodies in the country to have a first year open field trial. So we have a first year open trial for BT transgenic and PGNP. We hope that once this Brassica, Juncia, the transgenic will be cleared by regulatory authorities, this will pave a way for transgenic PGNP and chickpea as well. Similarly, in crops like, the urad bean, mung bean, where the YMB resistance is required because the crop in India is grown in a different place, in a different seasons. So YMB is a big problem. There again, we are making efforts to combine resistance for three different, I should say, very distinct strains that is available in the country. So this is very brief about the product which we feel they are in the pipeline. And once this product will come in the market, perhaps it will make a big impact and further, refine our breeding protocols. We are using, the information bioinformatics tools to modernize our breeding program, digital platform, with ICRISAT as one of the partner. And we feel that plant breeding will be more, rewarding in time to come. This is all I wanted to share, Chairman, sir. Thank you.
Thank you very much. Yes, could you give him the mic? We now invite from China, Dr. Boshu, to make his introductory remarks. Maximum time, speaking time is 10 minutes, please.
Thank you. Good afternoon, everyone. So I'm from the Oil Crops Research Institute of the Chinese Academy of Agriculture and Sciences. The location of our institute is in central China. I'm basically a peanut breeder. As Rajiv said, my institute have had a very long history in cooperation with ICRISAT. We offer cooperation at least 35 years now. And it is my honor to be here at this time to attend this meeting. And I would like to say it again, we sincerely congratulate, express our sincere congratulation to the genomic research progress achieved during the past 10 years with the leadership of Dr. Rajiv Vaishnavi. To my understanding, the innovation directions of crop science must include at least 4 components. I'm not talking about the ICRISAT demanded crop of my crop only. Basically, I mean, in general idea for all crops, I think the 4 components are the same. First thing, how can we increase the yield potential of the crops to increase the stock price in markets? Secondly is how to improve the quality of the crops to meet the need of changes nutrition demand as well as industry. The third one is how can we guarantee or ensure the safety in production, the safety of production, or production safety, environmental safety, and as well as food safety. Lastly, we should think about how to improve or how to enhance the production efficiency in farm field. Nowadays, in countries like China, even India, the manpool for agriculture is somehow a problem. So our taking knowledge, the crop production should, attention should be paid to how to decrease the cost of the manpool to enhance the production efficiency as well as to increase the farmers' income. So I think my idea is we should think about for the future the 4 components of innovation directions. China, as you know, is the large country in terms of agricultural production, as well as China is also a very huge market for agricultural products. China is also a growing country in terms of crop science, including the genomic research. And to my understanding, maybe not all, as Dr. Binghang is here, and we have still some Chinese scientists attending here. As I know, we have contributed much to the genomic research of rice, cotton, wheat, rapeseed, cucumber, sesame, and even microbe, peanuts. During the past 3 decades, the production of most crops in China increased greatly, in which the variety improvement has contributed greatly by using conventional and the molecular approaches. For example, my crop, the peanut, it's also an ICRISAT demanded crop. It's grown in about more than 100 countries in the world. So in China, during the past 3 decades or 35 years, the production of peanut increased dramatically. Based on the data released from FAO, the production of peanut in China in 1980 was about 3.8 million tons. Now, currently, the production is about 16.8 million tons. That means the current production is 4 times more, 4.5 times compared to that in 1980. What contributed to this kind of a change? Basically, it's a variety improvement. When we compare the average yield of the varieties, peanut varieties released in this year, I mean the current time, which is at least 2.5 times of the varieties we grown in 30 years back. So we believe that genetic enhancement for the crops has been very important. However, the current progress is still not enough. As you know, further genetic enhancement for most of the crops could not be achieved without extensive utilization of the advanced technologies based on genomic research. So during these days, we are talking about genomic research progress and the potential utilization, and I learned a lot. In the aspect of genomic application and perspectives, the peanut breeder and a director of our National Agricultural Research Institute, I have just 3 suggestions. Firstly, we should carefully propose the goals of our further breeding programs. The further breeding objectives should be of a high standing, challenging, but achievable. We must be clear that for what kind of traits, molecular markers, assistance selection, or genomic selection of gene editing techniques could be used. In the peanut, we hope ICRISAT and even other organizations could further work on genomic approaches for efficient selection for specific traits, in particular for specific fatty acids, soybean disease resistance, in addition to the available achievements for foliar disease resistance and even the drought tolerance. So this is my first suggestion. Secondly, as a national organization, we should not just wait for the coming of advanced breeding technology from ICRISAT or other organizations. We would like to work with ICRISAT and other international partners to develop efficient and useful genomic tools. For example, in my institute, we would like to work more on resistance to bacterial weight, steamer rods, and even resistance to bacterial weight. I believe this kind of participatory innovation would be necessary both for promoting the technology development and enhancing capacities in the developing countries. Lastly, we would like to try our best to be prepared for the ongoing transfer of genomic-based breeding techniques, including being prepared in breeding populations, being prepared in necessary facilities, and being prepared in mechanism of extending the improved varieties and other products to farmers and the concerned industries. So these are my 3 suggestions. Thank you very much.
Thank you. Thank you very much. Before the next speaker, may I invite Dr. Arvind Paddy to join the panel. Thank you. So, Paul, over to you.
Oh, thank you. It's a great honor to be in this conference, and I will continue thanking Rajiv for inviting us to be part of this panel discussion. I represent Africa, and you all know the application of genomics in Africa is still fairly low, and therefore most of the breeding programs, are struggling to begin to adopt application of genomics. And therefore, I would use one example that we have done in our university in collaboration with ICRISAT. Some 3-4 years ago, we were involved in Generation Challenge project where we were introducing root QTL traits and yield into our Kenyan chickpea lines. And this was a collaborative project that was also involving some partners in Africa. Ethiopia was one of them. And the experience that we got through the collaboration has given us a sense of possibility that the NAS in Africa can deploy genomic resources in their breeding program. Because when you look at all the programs in Africa, majority of the NAS in Africa are just still doing conventional breeding. And the integration is actually almost like a must, because we are seeing quite a number of challenges coming up, especially with climate change. We are seeing new diseases. And the latest challenge that we have seen now is the fall armyworm, which started in Southern Africa. It has come to Kenya, and it is causing a lot of damage. We think application of genomics could easily solve or reduce the incidences of diseases and pests in such situations. So in this case of chickpea, we started by looking at our elite lines. We did SNP marker analysis, and then we were sourcing the genotyping from K Bioscience in UK and also in ICRISAT. Finally, after 2 years, we now got products, MABC lines, which are good. They have 10 to around 40% better yield than the parents. We are currently using the same material to improve our varieties, and we were quite happy. I was quite happy to come and realize that there is a project now, the ATBT project, that is coming up, and we would also wish to be part of that so that we can improve the application of molecular markers in our breeding program. Because as a routine application of molecular markers in our programs, it is, I've said, it is quite still scanty. As we look at the need for genomics in our program, there are quite a number of other success stories that we've been involved in cassava, where we had collaborations with Becker, and we developed very good lines that were resistant to cassava brown streak disease. We looked at good sources of resistance from a variety in Tanzania, and large populations were developed, and we are currently seeing reduction in this emerging disease that was minor a long time ago, and it has become a major disease. And therefore, use of genomics is actually assisting the NAS in Africa to reduce the spread of disease that was in one region in the coastal areas, and it has emerged as a major disease in the highlands. We have also seen the use of IR maize from CEMIT. We're involved in a Semlensa project where we discussed with Prasad, and he told me there are markers for the IR maize, where maize is coated against the striga weed. And striga is also a major weed. It is causing a lot of significant yield losses in most of the African countries. And from our observations that we've been doing to deploy the IR maize from some seed company, we've seen very good reduction in the incidences of losses caused by striga. We think if such technologies can be developed and incorporated in other important crops like rice, sorghum and finger millet, there is a possibility of enhancing biotechnology use and also encouraging integration or incorporation of genomics in our breeding program. Because the NAS partners are not looking at technologies. We are measured by the number of varieties we give to our farmers. And therefore, we actually are always looking at solutions that can offer quick food security issues, because that is the major challenge in Africa. What we have realized is that the technologies change very fast. Before the SSR markers were deployed in Africa properly and adopted, we are seeing SNP markers, genome-wide selection, And therefore, the technologies are changing very fast, and sometimes it's confusing to NAS partners. But what we normally say is that we better focus on the problem than the technology. There is potential to adopt genomic application through collaborations, through proper preparation of genotyping facilities, and also genotyping facilities in our region. And this would actually, we look forward to enhancing collaboration with CEG centers and international institutes so that we can be able to adopt the genomic tools in our breeding program. Thank you so much.
Let me now invite Dr. Khaled Amiri from the Dubai Center. UAE Center, sorry.
Everybody says Dubai. That's the, okay. Well, good afternoon, everybody. I would like to thank Rajiv for inviting me to this very informative symposium. And this is my first time in Hyderabad, and I already feel at home. Now, I come from UAE. Obviously, UAE is facing a lot of sea, and to our backs is a lot of desert. So our environment is not as friendly to agriculture. But the country has actually invested a lot in agriculture in the remaining areas that we have that is plantable, and since 1970s. And by subsidizing the farmers, by providing lands and so forth and so on. But still we are faced with three elements, environmental elements, which is we have water issue, it is hot, and it's salty. So in our center, in Khalifa Center for Genetic Engineering and Biotechnology, which is about 42 years younger than SIG, we are not studying, we are studying some crops, and one of my colleagues is here, Dr. Martin, he's sitting right over there. And we are working on transgenic, but we are also studying desert plant as a model for abiotic stress tolerance. We have been working, we have actually sequenced the genome of prosopis, which is also legume, and we are trying to generate enough information and knowledge to be transformed or translated into improving crops that are amenable to live or to survive in abiotic stress environment. So in Khalifa Center also we are doing that by extensive uses of genome, which I think is the future, is a commodity that will come one day. And also we are also studying GWAS to study other crops or plants that actually, that are, some of them are native to these areas, which is palm, date palm. And we are using GWAS to study different character or different traits. We are also developing some, we have discovered some of the genes or identified some of the genes that we will use to transform trees, whether through transgenesis or through mutagenesis. And this is what we are actually involved in the center at this point. And hopefully we're looking here also, which thanks to some of the colleagues who have actually provided good information for us. And we are actually working, trying to also develop collaboration, as I think science is an international effort, and not only a national effort. And for our center, that's what I have to say. Thank you.
Thank you. Now, we have someone from the FAO who I believe can speak for Africa properly. So over to you, please.
Thank you, Eric. Good afternoon, colleagues, ladies and gentlemen, friends. The downside to intervening late in the program is that virtually everything you say has been said. So one begins to sound kind of hackneyed. But Rajiv, Anu, the Center of Excellence, and ICRISAT, I hope you haven't been thanked so much that you consider my thanks for this opportunity to be here as being worthless. It's from the bottom of my heart personally. And when I was introduced, one thing that Rajiv mentioned is that in a previous incarnation, I had worked in CIAT, the Centro Internacional de Agricultura Tropical, one of the CGIAR centers. What he left off mentioning is that in my part of FAO, we almost look like a commune of CGIAR researchers on some form of extended away without official leave. That is because our Assistant Director General, Ren Wang, used to be the Deputy Director General of IRI. He was also the Director of the CGR in Washington, D.C. in the World Bank. Our Divisional Director, that's the Director of Plant Production and Protection Division, Hans Dreyer, is also ex-IITA. So you can then imagine the kind of message that they sent. You know, they want me to come back and report on how the deliberations here are going to contribute to our efforts in assisting member countries, as has been severally alluded to, achieve the Sustainable Development Goals of eradicating hunger by 2030. As we all are aware, this is a very ambitious goal, considering that even as we speak here, about 1 billion people go to bed hungry every day. About double that number are malnourished. And FAO takes these problems quite seriously. By modest estimates, we are supposed to produce 60% more food. And this is in spite of climate change. This is in spite of the fact that we know there are no more redundant natural resources to deploy. We don't have redundant arable lands. We don't have redundant water resources. We must produce this significantly more food without further damage to biodiversity, without further damage to the environment. So how can this be achieved? FAO believes that all options are on the table, including biotechnologies. And to that extent, genomics as well. So what are we doing in this regard? We hosted a global symposium on biotechnologies early last year, and the consensus was that biotechnologies are contributing to aiding family farmers feed their households and achieve better livelihoods. We are proceeding to having regional meetings. We have had the regional meeting on biotechnologies for Asia and the Pacific in Kuala Lumpur, Malaysia, end of September. And Rajiv, we reiterate our thanks to you for serving on the advisory panel and for ICRISAT for letting him participate. We've also had a regional meeting for Africa barely 2 weeks ago in Addis Ababa, also trying to get the perspectives of grassroots, the stakeholders who we serve. Again, a member of the CGIAR, Melaku is somewhere here, from IITA, served on that panel. A number of individuals from the CGIAR also participated. So biotechnologies are important. Now, if I may throw out a few posers to us here, we've been talking about genomics, and genomics implies the generation of massive amounts of data, the so-called digital sequence information. Now, the way it's being presented here, it is as if all is well. We will just willy-nilly have access to this body of information. I'll get back to this in a moment. But another topical issue that we hear about a lot now is synthetic biology, the ability to synthesize proteins, genes, and according to some people, even whole organisms. We're also hearing about genome editing. That has been mentioned quite a bit here. And genome editing, interestingly, is usually conflated with gene drive. Some people even, mention the two as if they are interchangeable. Ladies and gentlemen, it's not all rosy. Not everybody shares in our optimism, in our enthusiasm about these advances in biotechnologies and in particular genomics. In Cancun in December, during the 13th Conference of Parties of the Convention on Biodiversity, at times it assumed a kind of warfare type of environment. Some people were calling for the moratorium on genome editing, conflating it always with gene drive. Some we are also now looking at the possibility of having some access and benefit sharing regime regarding digital sequence information. So I think we should all be concerned about how the work we do will ultimately be used in advancing this cause of producing significantly more food to feed an ever-growing population in climate change scenarios. But we are not helpless. We are scientists and relapsed scientists like myself. We are not helpless. I urge all of us to talk to our national focal points to the Convention on Biological Diversity. You don't have to tell me what your impressions are, but do talk to them so that when they attend these meetings, they represent you. The same for the focal points to the International Treaty on Plant Genetic Resources for Food and Agriculture, to FAO's Commission on Genetic Resources for Food and Agriculture, so that we do not make the same mistakes as continue to bedevil our efforts with genetically modified organisms. So, ladies and gentlemen, these are some of the thoughts that I want to share with you. And while I welcome the receipt of perspectives that I'll go back to Rome with. Thank you.
Thank you. I think we now want to bring in the private sector. Dr. Victor Nwosu from Mars Incorporated.
Thank you. First of all, like everybody else, I'll say thank you to Rajiv for inviting all of us here. And I'll say thank you to all of you in the audience for being here to participate and listen to what we have to say. Before I start, I'll go a little bit to let you know what my perspectives are and what shaped those perspectives. I work for Mars Incorporated. Mars Incorporated has chocolate, gum business. We have the largest pet business in the world, and we also have rice business. So what does that mean? We see ourselves as an agricultural company, not so far as a manufacturing company. So with that in mind, you also have to know that we're a privately owned company. Why is that important? That is crucial to what forms the perspectives I'm going to share with you today. We have this view that we are the interface between the consumers, the research, and the people who supply the ingredients. And that's very, very crucial because we see things happen and we want to participate in it. The case in point is we had cocoa issue in Brazil. We had to work to get that solved by sequencing the cocoa genome. It was a privately done thing. And for that to happen, we also knew that we have to form an unusual alliance because sometimes you just have a some view that you can only work with certain people. So I borrowed a lesson from that, in that if you have these ideas on how to feed the world, which is what everybody has been talking about, Dr. Singh started by saying the product. You are working on a very, very crucial technology. And that technology is very crucial from my point of view, in that it is the way we can feed the ever-growing population. All right? And it is the only way we can sustain the crops that will feed the people. And Dr. Jones started the other day and saying, well, you developed one resistance, you may create other ones. So, which means all these things are dynamic. So we cannot adapt without investing in technologies that will help us keep the crops sustainable and keep things going so that we can feed the world. So with that said, it is important that when we do this work, I saw most of the measures were in publications. That's a measure of good quality work. But the end product is the material that will feed the people. So if you have that in mind, it's easy to have a value chain viewpoint to what you do. And you are very, very crucial to all these things. So what does that mean? You have to listen or connect with the farmers, connect with the suppliers, connect with the end users. And therefore, you have a completely clear picture. What you are doing, you are contributing a whole lot. And sometimes you undersell it. So why is that important? We've mentioned gene editing, GMO, and all this stuff. Why did we get there? Because we let politicians take over. Because we never did communicate effectively. But the opponents called it Frankenstein food. See, the key word is the language, the communication. How you brand what you do is very, very important. We should never take that for granted, because we are doing very, very vital, especially you in the audience here, are doing very key work that is the base for human existence, food. And you have to brand what you do and be proud of it. All right? So that's very, very crucial. And I can never underemphasize that once we look at things, we'll remember why we're doing it. It is to create food, nutritious food, that feed the people. So, is it important? Yes. It is one of the things that cause problems is food insecurity. And that creates some political issues. So, having secured food system, good quality and nutritious food is very, very important. So, to me, that's my perspective. Keep focused on what you do, brand it, and be proud of it.
Thank you. Dr. Oscar Raira Lisa Razu from Dow.
Wow, I owe you $5. Anybody that can pronounce my last name deserves $5. Thank you. Again, like everybody else, I'd like to thank the organizers of the meeting for inviting me to come to such a conference. I've really enjoyed it. I think as some of my colleagues have said, it's been a very science-rich conference, and I've taken the opportunity to catch up with some of the developments in the field, since so my activities have kind of deviated a little bit away from that, and dedicating most of my time to managerial type activities rather than science. To tell you a little bit about me, I'm basically a new employee. As you guys know, Dow Chemical and the DuPont company have officially merged as of October of last year. And from that merger, we have what we call Dow DuPont. And within that, each of the companies have a number of, let's say, holdings or subsidiaries, and all of those are getting combined so that we'll have three independent companies that will spin out of Dow DuPont. So there's going to be a business that's going to be dedicated to material sciences, one that's going to be specialty products, and one that's going to be a pure play ag company, which is now the ag division of Dow DuPont, which will spin out to be in that independent ag company they talked about. The ag company is a blend of Dow AgroSciences, which was one of Dow's companies, with DuPont Crop Protection and DuPont Pioneer. Dow AgroSciences had both a crop protection component as well as a seeds component. So the blending of these three, businesses will lead to the production of an independent company that will, of course, focus on seeds, traits, and crop protection solutions. We view ourselves as a science-based company looking for solutions to contribute to the well-being of the entire agricultural system, from producers to end users. To give you a little bit flavor of what will come is, among other things, we think that the combination of the pipelines of the companies involved will be richer and deeper than is available currently. The technologies coming from the Pioneer side or the DuPont side and the Dow side are complementary but not duplicative, so you'll have more options there. We are still caught up like everybody else in providing insect protection and herbicide solutions on the transgenic side only because of the extreme cost of deregulation and all of the politics associated with that. And I will echo some of the words that have been spoken in that regard. It's a big issue, and if we could somehow lower some of those barriers, we could deploy more technology. And again, it's not for the lack of science. As you well know, it's some other issues that are a little bit more complicated and difficult to deal with. With respect to seeds, diversity. And I have to write this down because when you start combining both companies, we find that we work on corn, soybean, canola, cotton, wheat, sorghum, sunflower, brachiaria, rice, mustard, permillet, and alfalfa. All right, so that's what you get by combining two companies that perhaps have different focus, but now bring them together. You see the diversity of the activities that we're going to be engaging in. Okay. As far as the deployment of transgenic technologies, we will be doing it in corn, soybean, canola, and cotton. All other crops, they'll be conventionally bred, right? And some of these crops will be conventionally bred for a particular geography that doesn't take a transgenic technology, for example. Okay, that's one example there. We also do quite a bit of work on the pathogen side and the pest side. From the genomics perspective, we sequence microbial as well as pest genomes to better understand pest dynamics and disease dynamics and apply those to solutions that we're looking for. With respect to traits, not only do we have transgenic traits, but we also work quite actively in native traits. And of course, we use the platforms developed by the scientific community for gene discovery. And again, those will be targets for gene editing or trait development. And the native traits activity is very hot right now because, again, deploying transgenic traits is so difficult. So native traits, we think, may be a little easier to do that. Unlike other companies, we've also been very active or interested in what we call output traits rather than input traits. An input trait is a trait that you use to protect your crop, for example. Herbicide resistance and insect resistance will be input traits. But an output trait really is what the end user benefits from. It's changes in quality or nutritional aspects of the crop that comes out. One example of an output trait would be omega-9 canola that we've worked quite a bit on. And it's what we call a healthy oils product and output. We have an engagement with Arcadia Biosciences, a company that sprung out from the Tilling technology situation. And we're interested in developing a quality trait for wheat. So then those are some of the differences with respect to the synergy that comes from bringing companies that have science-based offerings that are complementary rather than duplicative. with respect to the application of technology and in genomics in a specific, we are extremely encouraged by the implementation of genomic selection. And we have seen that indeed it's, with respect to some recent data, that it's accelerating the rate of genetic gain in our breeding programs. So for the large-scale breeding programs that we have, genomic selection is basically completely intertwined. We do it and we'll continue to do it. It's not a question of if, it's a question of when and how, because not all crops are the same. Obviously, corn and soybeans are king crops, there are core crops, and then all these other crops, of course, take subsequent, secondary positions to that. But in any case, if we can and it makes sense to do it, we'll definitely deploy that technology in that regard. I think I'm going to stop there. Thank you.
Thank you. We now invite Dr. Mike Butterfield to make his remarks.
Yes, thanks very much, and thanks to Rajiv and the organizer for the opportunity to be here today. So as Rajiv mentioned, I actually work on sugarcane, so I feel like a bit of an outsider here because it's more of an industrial crop as a crop that, we're talking about, feeding the world, and, sugarcane is now getting a bit of a bad reputation in terms of if you eat too much of it, all the things that can do for you. But just to explain a bit in context about what I will be explaining my perspective. So as Rajiv said, our company, we in fact are a private company, and we like to say we're the largest private sugarcane breeding and research company. And that's because, in fact, we are the only one in the world. Because if you look at sugarcane breeding worldwide, it's either government-funded programs or industry-funded programs. And our company, which actually started out as a cooperative 40 years ago and went through various changes, now has been spun off as a company. So we survive basically on charging royalties for the sugar mills that grow our varieties. And our competitor is the national program that charges nothing for their varieties. So you can imagine it's a pretty difficult business model. But despite that, we are moving ahead, and in fact, we'll be listed on the stock exchange in 4 years' time. So anyone that likes a risky bet, I'd invite you to buy shares in CTC. And I think really that is where I want to bring my perspective in terms of genomic applications. Because if I go to my boss and I say to him, I want to spend $100,000 on doing resequencing of my varieties so I can understand how the variation is impacting on traits, etc., he will say to me, okay, well, what's the cost benefit of that? What is the value of a genome sequence? Because for him to approve that spending, he has to show that there will be some kind of return. And it can be a bit of a double-edged sword, because if I say to him, okay, well, by doing this genomics work, we can improve the efficiency of the breeding program, then he will say, okay, you've improved efficiency, therefore you need to either cut your operating expenses or cut your people, because if you've improved efficiency, you're doing things equally for less. And so really, I think that's the challenge that we have. So as scientists, we really know that the basic tools that are being supplied in terms of all the genomic work that's been doing really has a benefit. But to quantify that benefit becomes very difficult. And so I think I'd like to throw that challenge out to the audience. So if you imagine that you have all bought shares in CTC, I come along and say, as my shareholders, I want to spend $100,000 on doing PacBio, 10x Genomics, HiSeq, so I can have a really nice genome sequence so I can start looking at my data. How much money are you willing to invest for me to do that? Because really that's what it comes down to, is from my boss's perspective, if he has, he is looking, am I going to spend $100,000 in investing in genomics technology, or am I going to buy a new tractor, or am I going to do an improvement to a building, or am I going to create a new sales and invoicing system that will help with my cash flow? Those are the kinds of day-to-day decisions that he faces and that we have to justify to him in terms of, in fact, how we use all of these genomic tools that are being developed. And so, yeah, I think as we move forward a bit later in the discussion session, I'd like to throw out that challenge to all of you: is how do we quantify the value of a genome sequence or a resequencing panel? Thank you.
Thank you. We bring in Dr. Arvind Paddy.
5 minutes, okay. Can I stand for a while, please? I just have a problem with my back. Thank you so much. Rajiv is not there, but I wish to thank him for not for putting me here, because I have spoken in many podiums, but Rajiv has given me the opportunity to reunite emotions. It's a pleasure to speak before my guru. Dr. R.B. Singh, the co-chair, was my director when I was a student in the IARI in 1996. Dr. R.S. Paroda was the Director General then of the National Agriculture Research System. And also it reunited with some of my old friends. Niranjan, can you kindly wave your hands, please? I met him after 30 years. And Sujay Rakshit is there. He was my junior, 1 year junior. Gangarao was my classmate again. And many of you are very known faces. In fact, I was speaking with Gangarao and we met Professor Gupta. And we told that, sir, aapke kitab padh ke hum log IAS bane hain abhi. So I was very grateful to each one of them. And this has given us a very unique opportunity. But I will speak sense for 5 minutes as I committed to Eric. Because though I am a part of the ICRISAT system for now, I will go back to the Indian Civil Service after 2 years. I have just come on a lien to this organization for 3 years. But I will try to give my perspective as to how science could be linked to policy, without which maybe the end outcome or the fruitfulness of research has no meaning. The research outcomes have no meaning if it doesn't reach the farmers, the poor farmers or the smallholders or the tribal farmers or in South Asia or Africa, anywhere. I'll just give you a very good, interesting story. I'll confine within 5 minutes and you'll like it. And I'm speaking from my own experience. It is a scientific story, you can say. When I was the commissioner or the director of the department, the agriculture department, fortunately I have a background in agriculture science before joining the Indian Civil Service. I was drafted to the International Rice Research Institute in Manila for a very short training. There I met David McHale, the one who was heading the STRASA project, the stress tolerant research for Africa and South Asia, right? Okay, STRASA project. So he introduced me to the science of this sub-1 QTL. He tells me that, this sub-1 I know little bit of science, though not much as you all would be knowing. He tells me that this sub-1 QTL was derived from a land race in Odisha, my home state, the FR13A. Those of you who would be into rice science would be knowing it. It is a very familiar term to all of you. Then he tells me that as to how this could revolutionize the agriculture scene in many parts of the eastern India, because there is a mega variety called Swarna in my state and many adjoining states. And then he says that this could tolerate the submergence for about 12 days. And then I go back with that perspective to my home state, and then the experiments had already been done. I told in the beginning that I would speak sense because I want to link science with the policy making. And when we go back, then the research the experimentation had already been done, that it is literally the sub-1, the Swarna sub-1 variety, new variety, that can withstand that can tolerate submergence to about 12 days or 2 weeks. And then the policy making starts. Then the local university would be called. Then there would be a committee at the state level. then everybody would speak, no, we can't release a variety unless and until there has to be, and there would be some kind of multi-location trials across the different agroecological zones and all this stuff. Dr. Singh, you would be very familiar with this when you release the varieties from the ICA system. Then I would tell the secretary then, I was not a secretary then, I was a young officer, 10 years older, younger than what I am today. And then I would tell the secretary, sir, don't teach us any science, and nobody should teach us science anymore. The scientists have already proven that this particular gene is responsible for tolerance. There has been a lot of investments from the Gates Foundation and other like-minded partners, and the result is already seen. If you suppose try it for 2 more seasons in multi-locations, what extra would you gain? And in the process, we would miss out releasing a variety in the interest of farmers. You can imagine it had only 1 season trial. It is in the history of maybe release of any variety in our country that I did not use my veto, but I used my influence in the decision-making space. And then we released, Odisha, or the state that I represented then, was the first state in the country to release Swarna Savan then. And now it is such a huge success. And who is reaping the benefit? It is the poor farmers, not only in India, Bangladesh, across the region. So if we don't link policy space to the scientific discoveries positively, and we don't sort of make the so-called decision makers appreciate the research, the toils that those have gone into a discovery, it is very difficult to make the outcomes, the scientific research outcomes, reach the farmer. The story is linked to another one. Then Mr. Bill Gates would come to India and he would invite few select people to his suite in Delhi. And I was one of the few those who were invited, including Mr. Subba Rao, the one who introduced IR8 in our country, and Subba Rao, the IR8 man, the miracle rice man, and few others were there. And then nobody would dare take a picture with Mr. Gates then. Then I would tell a gentleman sitting there, then this is my mobile phone. Can you kindly take a picture of all the persons whom Bill had invited then? And then that gentleman would take the picture, and then you can Google. I go by Arvind, though my name is spelled in Sanskrit, it is like this. But I go by Arvind, but you can Google Bill Gates and the stuff. And that very gentleman who took the photograph, can you take a guess who he could be? Yes, please. I've given the clue already. It was none other than the present DG of ICRISAT. He was with the Gates Foundation then. So I told David, my team lead now, that it was you who took that picture then that became a very memorable picture for me for the entire lifetime. So I just would sum up here, and I would like to drive the point that all the scientific discoveries need to reach the farmers' fields. And how do we make it reach? We need to sensitize the policymakers, both in the National Agriculture Research System in our country, it could be ICAR or Ministry of Agriculture, but also in the state agriculture universities and the space. I thank Rajiv particularly for giving me this opportunity, and thank the co-chairs for this time. Thank you.
Yes, thank you very much, Arvind. The time has come for us to open this discussion, but before I do that, I would like to perhaps make a couple of comments and direct the discussion. From the submissions that we have heard, I think we can all conclude that they are telling us that innovations is what will bring about the inclusive transformation of agriculture. But our friend from the FAO did caution us about technology, because people are careful about what they embrace. So another dimension is education. I think innovation and education is very crucial. If we listen to the talks in the morning, we heard Dave Edwards talk about, if you like, technology and how he's optimistic that Caspar Cripps will if you like, do all the wonders that we need. But again, we have to take this with some caution. We're warned about switches which can turn on lights and also lifts. And we don't know what will happen. For example, we're having a conversation the other day when cassava came on the board. I don't know whether you know cassava. Someone said that this crop, one time I read that in Africa, where the most important staple is a very bitter root. And I felt very sad, though, because it's a delicacy. How do we knock out all the genes in cassava which make noise? What would be the consequence of knocking out these genes? We don't know anything. I believe it takes only advances in science. But there are challenges. For example, how do we address the gap? In Africa, the Green Revolution were lost. Apart from the Green Revolution, several developments, biotechnology, Africa couldn't get on board. Now genomics, gene editing is coming on board. What are we doing? I believe that CG centers are obligated to ensure that they pull the NAS along, because you're only as strong as your weakest partner. And the partnerships are very weak. You have advanced institutions in Australia, they tell you we are going to East Africa, we are not coming to West Africa. Why does geography divide us? We must think in the context of the Sustainable Development Goals, food security, ensuring that we end hunger, sustainable agriculture. It has implications for so many things. How do we ensure that every graduate student receives the kind of education that can make him or her stand his own anywhere in the world? We haven't gotten there yet. My friend from Egerton spoke about the problems in Africa. It's all about infrastructure, capacity. The human capacity problem has not been dealt with very well, and I think it's time that we took it head on. Today, if you talk to foundations, no one is interested in human capacity building. I don't understand. I say oftentimes that governments in Africa are sleeping. It's governments in Africa which must ensure that capacity is top of the agenda. I wonder what the FAO is doing, but I would like to open the floor for a conversation on how we can ensure that there is proper coordinated collaboration which ensures that genomics, which is an important part of the toolkit that we need to improve the crops that we so need urgently in the advent of the challenges that we face, is taken very, very important. So how do we move this revolution forward? How do we ensure that there are strategic partnerships addressing the problems that challenge us today? Any contributions from the floor?
Thank you very much. Sir R.B. Singh, I will take about a minute, not more than 2 minutes. You see, I've been here for last 4 or 5 days. I attended the meeting on forward breeding and then this meeting on genomics. I was expecting that there should be some connectivity between the two, but to my surprise, we are not talking about forward breeding in this meeting. You see, when you are talking about poverty, hunger, sustainability, they are the buzzwords which we keep on talking about. But the main point which we should perhaps discuss and consider, what traits in which crops are important. And to my mind, one is yield should be improved to meet the challenge of the population growth. And the other is we always talk about food security and nutritional security. So other than yield is the nutritional value of the crops. These are the two major components of the crop improvement program which we should address. In the yield, we know the major traits which are limiting the yield: biotic stresses, abiotic stresses. We also know which biotic stresses and which abiotic stresses. Then perhaps nutritional efficiency of the crops so that the fertilizer requirement should reduce. This is another important area which should be addressed. And then in the nutritional value, perhaps protein content, microelements like iron, zinc, carotene, and so many others. And in order to address this, we should perhaps discuss or consider to what extent genomics will help. And this is what this session is about. The genomics, when I consider it seriously, you see there is a word genomics assisted breeding. I don't like this word. Because genomics perhaps directly will not help in breeding. Genomics is a resource which will be used in breeding, and therefore I also thought that there can be short-term gains from genomics and there can be long-term gains from economics. Whole genome sequencing, resequencing which we are doing, this will provide long-term gains to the plant breeding program. Therefore, nobody would deny that it should be done. Short gain use of genomics, which I can think of is perhaps gene discovery is one. You can discover the genes for important traits which are not known through bioinformatics and genomics. You can perhaps use it in genomic selection, although genomic selection can be done without genomics also, because if you have a large number of markers available, even without sequencing the genome, you can undertake genomic selection. We have been discussing about it. But genomic selection is also not a part so far. We were discussing in the other meeting, forward breeding, high throughput genome sequencing, genomics. So this genomic selection, of course, to some extent, we think that can be used, but many people doubt it, that immediate future. They take, the time is not ripe. It may take another few years before the predictability of genomic selection would really come to the level when you will be able to use it. But the time perhaps is not far away. Within 5 years, we should be able to catch up, and genomic selection will become an important tool for the purpose of plant breeding.
Okay.
And gene discovery is perhaps, these are some of the, you see, you will indirectly use genomics for the purpose of discovering genes and other parameters which will be useful for plant breeding.
True. And good point, Mike. Good point, Mike. Thank you, Professor Gupta. Any other comment, suggestion? Yes, please.
Thank you, and I also want to thank the organizers of the conference and for the invitation to be here. And it has been great, this fantastic science that is going on, and we have seen that there's a lot out there that needs to be translated into gains in the farmers' fields. And from all the talks around, I see that we all have thoughts towards that direction. And what we need is to have all stakeholders on board to translate what we know in science into the farmers' field. So I am sure it's not by design, but I would say that for us to bring all stakeholders on board, we need to lead by example, and I would want to say that we would start by bringing some of our the mothers of our children on podia like this together with the fathers of our children so that we can ensure food security. And I can see that on the podia right now, we're quite gender insensitive.
Sorry.
Including the whole workshop. So I think that's something that needs to be considered, because if we have to raise food production, and I know FAO also knows this, that family food security is ensured by family farming, and if the women in the families are not included in all this, then I think we're running short of something. Thank you.
Thank you, Madam. Very good point made. As a matter of fact, after we started talking, I looked at the panel. I said, yes, probably a big mistake has been done. We apologize for this. But let me tell you, Madam, if this blue sky is held by 100 hands, 51 hands are women and 49 hands are men. And you hold the sky. And we will certainly follow you. And I would like to see that it is ensured. So we'll take note of it, please. This is humanizing science. We recognize that. Yes, sir.
Thank you, panelists, for sharing your wisdom on respective area of your expertise. My question is to friend from CTC. I wanted to know the status of the commercial approval of drought-tolerant sugarcane and high sucrose sugarcane in Brazil by CTC, and also other traits in sugarcane, which is nearing commercial approval. And what impact do you see of those traits in not only in production, but also sugar trade in the world? Thank you.
Thank you for that question. Yes, so in fact, this year our company received commercial approval for commercial release of BT sugarcane. So resistant to stalk borer, which is a big problem. And that's the first transgenic sugarcane release in the world for commercial production. In terms of drought tolerance, it's not really part of our product pipeline at the moment. We have some collaborations with Embrapa and some other companies on terms of drought tolerance, but that's an experimental level, so we don't really have a timeline in mind on when and if a drought-tolerant product will be released, because currently our product portfolio for transgenic varieties is focused on insect tolerance and perhaps some herbicide tolerance in the future, but our pipeline at the moment is just insect tolerance. So the idea is to launch an additional 5 or 6 transgenic varieties also with BT, various forms of BT.
Very much. Brazil, the world leader, as a matter of fact, in many. I just wanted to add, it was in Brazil for the first time ever when the rhizobium-based sugarcane, or the whole rhizosphere-based sugarcane industry, was really brought to bear upon with the new science. Dobrena, the lady who really led the Brazilian development based on the microbes, and that microbial work that she did and the microbial genetics work later on is again the world capital of microbial development based particular industry, and that is Brazil. So I thought this is a wonderful coincidence to share that happy news. Any other question? Oh, sir. Please.
Thanks to all, and I am highly thankful to Rajiv Vasne sir. He invited me, and I have one question, very small question. As you know, first day Darwaje talked to, we have our Narendra Modi have target to double the income in 2022, farmers income 22 double, and it is very big challenge for us to double the farmers income in next 5 years double. And that's why I'm asking, whatever the advanced science is going on, and every year new science is coming, and these science can double the income in next 5 years to the farmers.
Yeah. A big answer to that you asked. The whole country, discussion, debates going on. But one thing is very clear: if we have to really double the income, we have to increase the productivity, genetic gain. And for genetic gain, you need in a kind of science like genomics, that in a very short period of time you can increase the gain. Another thing is how to bring down the cost of cultivation. That is another thing. The third thing, how to go in a sustainable way, not destroying the soil, not compromising with the environment. And at the top of everything, the farmers have to get a good price. For that, we need a policy support. That is most important. Because unless there is a policy support for getting a good price of the product, income will never be doubled. So these are some of the clues, that we are trying to compile and put up to the, the committee that has been constituted in India. Thank you very much.
Yes, yes.
Sir, before I speak, I would like to second the lady who proposed that this is not a panel, this is a manel. In fact, Professor Paroda was there. We were there in a conference, and then we attended that conservation agriculture conference, and all were men. And there was a very stinking sort of, and a very positive reply from Ranjita, the gender expert in IRI. And she writes that, Arvind, as a friend, I have a request that you don't be a part of any panel where there is no lady member. So I, in principle, support your idea that at least we have so many lady. Sir, I'm sorry, I'm disagreeing with you. You have been my teacher, but this is literally the concern everywhere, that any panel should also have a lady member if there are experts in the audience. This is one. Number two, on the on the doubling of farmers' income, which is very contextual to Indian scenario, that our Honorable Prime Minister has told that by 2022, the farmers' income should be multiple times, at least not double. We should not stay there. But as genomic scientists, as all of us agreed that we are into a long-term goal, but we have a big contribution to make. But productivity, and as a policymaker, I would say, productivity would not raise the income. It is the profitability part, which Dr. Singh also mentioned, that we need to link our production to the markets. We should de-intermediarize the entire supply chain and the value chain. Then only we can, but that's a big debate. But that's my humble request, sir. I'm disagreeing with you that we need to have a lady member in all panels that we organize anymore.
If you can have.
If we can. Yeah, that's
sure.
Right.
Victor, you want to?
This is a little bit in the response of the last question. We should have in mind that some of this technology is new. It's not quite that old. But what I can tell you is right now on this campus, Rajiv here, we're working on addressing some of these issues that benefit the farmers. When I started looking at, we opened a facility in Pune, not too long, last October 28th. We started operation there, but ahead of that time, we wanted to get all our supplies of peanuts from India. So we wanted to know what are the issues the farmers face. And we realized it is disease, it is yield, all right, it's water. So we sponsored a project, Rajiv is not here, where he is stacking pyrimidine the genes for disease resistance because it costs a lot to spray. So you reduce input cost for the farmers, increase yield, at the same time give them some drought tolerance, not complete. So those issues are coming. But unfortunately, conventional way, it takes 15 years to commercialize a variety. But now with this technology, we can cut it down to 5 years. So within the next few years, you see some of those benefits for the farmers who are involved, right? And we are also buying from them. Maintain the income, okay?
Yes, please.
Thank you, sir. Sir, most of the time we are talking about the crop genomics for human mouth and nutrition. But animals are also a source of nutrients and important part of the farmer economics and important pillar for the farmer. And I think we should do our genomics for animals. And as morning, some scientists were talking about the personal nutrition. So now it's time to think about the nutrients for the animals. And so we can get a good income from the animals also, and we should think genomics for crop genomics for better health of animals.
Like I said before, we were working on alfalfa and brachiaria and some more forage crops, and of course the issue of producing varieties with high digestibility and high nutrient value is important, but it definitely addresses the need for animal side as well.
Yes, sir.
So my name is Christian Jung. I'm from Europe. And you may know in Europe, apart from gender issues, there's one most important issue today. This is climate change. I was a bit surprised that I didn't hear the word climate change. I think, except Dr. Ambar, who mentioned climate change briefly. So I would like to know how important is this issue in your countries? How would you rank this problem in comparison to the other problems you have? And second, how could genomics-assisted breeding help? Do you see special opportunities for exploiting genomic information to fight climate change or to breed crops which are more resilient to climate change?
I can start and then we can... I come from the US, and I think climate change ranks very high, even though that's not what you hear from some of our leaders. That's one thing. With respect to how we can apply technology to address the issue of climate change, as you know, there are activities in the scientific community looking for stress tolerance. We are interested in understanding the adaptability of varieties to specific environmental niches. We dissect the environment based on areas of adaptation. And we know that climate change is affecting what those zones look like, their size, and whether they're moving north or south, depending whether you're in the northern or southern hemisphere, right? And we are very much interested in also understanding what we call envirotyping, really understanding what the environment is and how to describe it, so that then we can couple that in variety development activities. So then we will have varieties that are targeted to specific environments or types of environments. We really didn't see anything about drones and satellite imagery and all of that stuff, which is another dimension that I know that our pioneer counterparts have invested heavily in that technology to really couple that with precision agriculture. And we see how all that meshes together with our ability to develop the best variety for the best environment under the best of conditions and also the worst of conditions. I think all of this kind of wraps around, and we haven't talked about it, but I think it's in our radar screen, right?
Thank you. At our organization, there are 4 cross-sectoral issues that we are tasked to demonstrate how our activities contribute to those. One is climate change. So we have to demonstrate how, for instance, I work in plant production and protection. We have to demonstrate how our work with countries supporting farmers in their efforts to transition to sustainable intensification of production systems, how that contributes manifestly to adaptation to climate change and indeed the mitigation of the footprints of agriculture that contributes to climate change. Another related cross-sectoral matter is biodiversity. We are also supposed to demonstrate that in producing more food with less inputs, we are not damaging biodiversity. The third one is nutrition. So it's not just gross yield. The food also has to be nutritious. And I alluded to the problem of malnutrition. The fourth one is gender. We have to also demonstrate that what we do is sufficiently gender sensitive to carry everybody along. And if I may pivot to the climate change issue, there is the Global Environment Facility and the so-called GEF, the 7th replenishment is Jeff 7. One of the impact programs is going to be food systems, and it is coupled to land degradation. A lot of what will be happening there will be addressing climate change adaptation and mitigation. There is also the Green Climate Fund, for which FAO is also an implementing agency. So we approach the issue of climate change from several dimensions. From the normative part, we participate, of course, in the implementation of the Paris Agreement, in supporting countries meet their so-called NDCs, declared contributions.
Yes, sir.
Thank you, Chairman, for giving the opportunity.
Yeah, I think I would also wish to mention something that is happening in Kenya on the development of GMO maize, the WEMA, the efficient maize for Africa, which is almost being released, but there are quite a number of policy issues, people fearing GMO. So there are quite a number of products that are actually targeting climate change, and also some that is looking at insects that are increasing because of climate change. So there are quite a number of GMOs that are in the pipeline, and they are almost going to be released. The government is looking at the policy issues on GMO because many of the citizens think that GMO has problems. So I think it is being addressed in Kenya.
India is a very diverse country. We have a very different kind of climatic conditions in the different part of the country and very distinct growing seasons. What we have seen over the years that there are increasing number of extreme events. And the precipitation pattern is also changing very fast. Like in northern part of the country during the winter season, earlier 20 years back, we used to get, the rain in December, January. Now it is very dry. And again, we see that in most of the crops, the season is getting truncated. I mean, season is becoming very short. Window where the crops are grown, that is becoming very short. And during the reproductive phase, we are getting, very high temperature, particularly for the winter crops in northern India. So these are the things that is happening. They have posed, a very different kind of challenge. Thank you.
Thank you, sir, for giving the opportunity. Sir, no doubt this genomics definitely will help in the crop improvement, both in the quality as well as the quantum jump in getting the yield potential of the individual crops, because it's more of a precision science, and every science in the history of the plant breeding has directly or indirectly helped in raising the food production from the tradition breeding to the mutation breeding to the polyploid breeding or the population breeding, etc., etc. Okay. Coming to the point that though we have used so many techniques for improvement of the crop plants, we have come up with several of the new high-yielding varieties and as well as the hybrids also. But till today, without any doubt, I can say that around just 25% of the area at global level is covered by the recently released varieties, that is newer varieties. So whatever the advancement yield levels we have increased through the other means of the our breeding programs, that not yet realized it. We have not realized that. That means the farmer has not yet realized it, which our Arvindo was just focusing on it. So we are thinking, just like private sector particularly, every 2 years or 3 years they are coming up with new hybrids, mostly in the market, and publicizing them. And 1 or 2 seasons they will be grown and they will disappear again. They will come up with another hybrid, private hybrid. Again, they will give some sort of the extension programs and propaganda, and whatever the seed that is produced by them, that will be sold and they will be getting the money. Everybody knows about it. What about our national programs, wherein the recent past, at least to take about 15 years back only, if at all we take it, how much of the area since 15 years, whatever varieties we have produced is under actual really, they are kept in the farmer's field. This has to be taken into the topmost priority because our concept of doubling the program, varieties are the hybrids which were released in the recently, about 10 years back or 15 years back, though the government of India has taken that no variety which is not released before, prior to the 10 years, should not be eligible for the taking some subsidy and etc. and etc. Even then, we are unable to make that dent because our varieties are the hybrids, whatever we have produced, they have the stress tolerance for both biotic and biotic also, high yielding potential also. But even then, new problems are coming, risks are coming. That's why in that case, we can get definitely the genomic science help and definitely we can end up with good results. But what is happening to the past science, whatever we have developed, past improvement in the crop improvement, what we have done it? Is it really kept to the farmer's field? No. Undoubtedly, I can say that it is no. Not even 20-25% also. So what is that we can say? Because here they has said that policy and the requirements of the people and as well as the farmers' income, rising of the farmers' income, it is the very most is the cost reduction technologies wherein the seed is the most important and very, very crucial component. Thank you.
Conveyed your point. He was very specific to India. He says the spread of the so-called released variety is rather not uniform, rather very, minimal and so on and so forth. He is mentioning that. I do not know whether similar problems exist in other countries. Probably they do. From Indian perspective, NP, would you like to say something? If not, then maybe I will say a few words on this.
It is true that many times, the variety don't reach to the farmer's field, or even if they reach, it is very late. But as you very rightly told, now the government of India has come with a program. If a variety is more than 10 years old, it won't get the subsidy. That itself is discouraging that particular trends. Now we see that more new varieties, they are coming up. Like say, for example, in pulses, we have a, new seed program where 150 seed hubs have been created. It is for less than 5 years old variety only. So what I mean to say, things are happening, but it takes time, before the variety gets adopted and it gives the benefit to the farmers. Thank you.
Yes, again, it will depend on which crop. This has to be taken as a differentiated, disaggregated approach. If you look at the BT cotton, 95% of the area in the country is under BT improved varieties. The contribution of both, particularly the private sector, is amazing. And the release of those varieties through the proper channel has been always very, very streamlined. And that continues because of the incentive that one gets about with the BT cotton hybrids. They increased sustained productivity, better return, good quality, and after all, the income enhancement. So these are the basic requirements if a variety is able to meet. 2, which crop we are talking about? Which variety are we talking about? As a matter of fact, if you talk of the self-pollinated crops, and in the self-pollinated crops, if you look at the replacement rate of the seed replacement rate in rice and wheat particularly, these two commodities which occupy almost about more than 60% of our total area over the 2 seasons, is absolutely 20-25% is the SSR. And for a self-pollinated crop, this much of SSR is enough. And the farmer seed program and the farmer safe seed program in India is also very strong, and that helps in the process. What is happening wrong at times and at places, that these quick fix processes of some of the states, of some of the intervention somewhere, do not allow to really strictly follow the rules and regulations already set by the government of India. Those rules and regulations must be. This is called implementation slippage. the flow of the valued seed, the flow of the quality seed from the breeder's plot to the farmer's field is already very well streamlined for different crops. It's just a matter of getting into. If you look at if you look at the crops and commodities, you will find in pearl millet the story is different, in sorghum the story is different, and so in vegetable crops the story is very different. So I will say don't lump everything in one. Look at the disaggregated way. Yes, there is room for improvement. It must be streamlined. The certification process and the distribution process, the private sector incentives, all those things have to be in place. But let me tell you, the seed sector in India is vibrant. It is responsive to the needs of people. It is always very adaptive to the situation. But then, of course, there is always a room for improvement, which you can always find here and there. But by and large, I will say that it's not as bad a picture as you are painting. I think we can look at it case by case, crop by crop, and look at the flow. Look the way the distribution of the pulses in 1 year, nowhere in the world. In 1 year, a pulses production in this country is number 1 today in the world. And if you look at the kind of 5 million tons added in 1 year, and the distribution of the seed available, which was prepared from this, was based on the flow from the breeder's plot of the known varieties. If this can happen in 1 year, 2 years, it can happen all the time. And I think it is just implementation slippage here and there. If it can be bridged, I'm sure it will be able to meet the requirements. So the point is not, we have digressed from the usual point. We are talking of the genomics application perspectives. And genomics can, of course, it is genomics plus. It is not genomics per se, but then let those genomically strengthened, augmented crops be given the environment from which they will be able to really explore the new possibilities. So it is plus the farmers' profitability, sustainability, productivity, all these factors come into play. Only then a variety will ultimately reach the right reach. So it is a combination of everything, which probably it is innovation plus. I think it is not just one thing, but the other environmental enabling mechanisms are equally at times much more important than the technology per se. So I thought let us keep these general questions out. And if you have any, we have another 20 minutes or so. If you have other questions related to the subject, please fire it.
Here, this is Prashant from Birla Institute, Jaipur. Genomics assisted breeding is a quite, often talked term, but there's a synonymous term called bioinformatics assisted breeding. I was just wondering, why isn't there a kind of a call among ICRISAT nations to ensure that we reach consensus in enabling, better crop improvement? So, for example, if we can infer certain genes that have come up, from bioinformatics analysis, right from in silico analysis to the bench to the crop, and reaching the farmers, for better crop improved products, could there be, a kind of a better consortia, so that, we can bridge that kind of, gap between genomicists and bioinformaticists? Is it because, the genomicists or the crop scientists, wouldn't trust the bioinformaticists, or what's really going wrong? Maybe I'm wrong, but I would like to just hear, the opinion of the panelists here. And the second thing, on a lighter note, it's unfortunate that there aren't women scientists on the panel dais, but I'm sure, the panelists are bringing out an opinion by thinking in the form of, women scientists. Thank you so much.
Any clarification? Well, if I may paraphrase, he was keen to know about how informatics has been or has not been able to really supplement your efforts in delivering something which could be useful to the farmer in the real sense. This is what is the...
I would have to say that's a misconception. I mean, when we talk about technology, it includes bioinformatics, information technology, databases, in silico assays. We know, and the community here knows, that if it wasn't because of bioinformatics, we wouldn't be doing the things we do with the data sets that we have. And yes, genomic prediction is done on a computer. And it's based on the numbers that are outputted that people are making decisions. So it's of course, part of the mix, yeah.
Bioinformatics is actually doing the whole thing. I mean, genomic information is available in the data, raw data. Everybody is, and through bioinformatics, these markers becoming established. So it is a bioinformatics base.
Yeah. Myself, Sunil, I have a question. I'm thankful for the panelists to sharing very valuable information from their programs. We are developing science at a faster speed, but we are not concerning about the extension of that science to the useful level. The development of a science is not a completion of a story. We need to make that science to be useful at commercial level for the product delivery. So actually, if we will think about the thousands miles away, there is a breeder in a small place who don't understand this technology and this information is generated. And he don't know that the information has generated in his crop that can be useful for him to enhance genetic gain in their programs. If he knows the information, the second level constraint is that he don't know how to use it. If he knows how to use it, he don't have resources to use it. So we should always think about that constraints and to make the faster speed of extension of this science, because it is imbalanced, like science is developing in a faster speed, but its extension and use at commercial level is at a slower level. So to balance these things, we have to always focus on that part also. I want the opinion of some of the panelists on this, and we have the policymakers also on the panel, so that those can also help to improve the extension of science. Thank you very much.
I am telling from the Indian perspective. If you look, compare, what was happening 10 years back and what is happening right now, I see there is a sea change. I know there are huge number of, that bulk SMS that is going, to the farmers on their mobile. There are mobile-based apps. Even there are hundreds of, that WhatsApp message, groups. I know. Every day my job is, 1 hour in the morning and 1 hour in the evening to see what is happening. So, and a lot of photographs, disease symptoms, things like that, it is happening. So what I mean to say, this IT tool, that has benefited to a very large extent. Now our reach to the farmers has gone many-fold more than what was happening 10 years back. So things have really improved, and there is more scope again to further improve it. There is a Kisan channel. 24 hours, the programs are running, experts are coming. So there is a radio and so many, things are happening. The extension has really improved, but there is still a lot of scope to improve further. Thank you.
Rephrase it, or you understand it now?
Yeah, basically.
Well, the question asked was...
my question was related to like extension of technologies to the breeders.
No, we got your, I think I am just trying to say what you said in a different way. Point is that there are people who are unreached. Can you reach them through the information system? That is what he is referring to. So it's as simple as that. Yes, you can.
Okay. Thank you. If I may provide some experience of what we are seeing and how we are intervening at FAO, there is a peer-led technology transfer mechanism that is known as Farmer Field School, which has been effective in helping extend the technologies, say, improve varieties and the production packages to farmers. So it's not a one-off transaction whereby an extension agent comes and talks about things and goes away. There is usually a demo or a series of demonstration plots where the farmers participate, and each community of farmers is led by a champion. That is one of them that had participated in a kind of training of trainer kind of program. Having said that, yes, there is no denying the fact that technologies are not reaching the farmers. In Africa, it's actually possibly even worse. There was, or there is, a recent study. The acronym is DIVA, D-I-I-V-A, Diffusion of Improved Varieties in Africa, commissioned by the Gates Foundation. It looked at 20 crops, new varieties of 20 crops in 50 countries in Africa. And the adoption rate, the average adoption rate is about 30%. And that includes all the major crops. So there is a problem there. Many extension systems, government extension systems are moribund. They're basically not doing anything, or they don't exist. So civil society organizations are contributing. Private sector is also contributing in advancing the technologies. Development partners like FAO using these demonstration plots and farmer field schools. So that is a constraint, but there are different mechanisms for addressing that.
Excuse me. I'm Bharadwaj from IRA. For these 2 days of deliberations, we see that there are large number of technologies on the shelf. The breeder who needs to use has his own local set of problems. In the previous HTPG workshop also, we have seen the number of interventions that are available for a breeder for a technology are very few, and they're not actually useful for him. Let's take of cassava, some of my friends were saying. The local thing that was actually responsible, for example, striga resistance, we don't have any marker technology. Now the basic thing that's happening is the developers of these technologies, be it the genomic tools or the SNPs validated, are developing and then asking breeders, rather than a tops-down bajai, it should be a down-to-top approach where the breeders are involved and made to understand. And then the second most thing that we are missing is yield, which is paramount for the farmer to use this. Except for Jivas, we don't have any other selection tools. Maybe any comments on this?
I guess the question is about how the genomic-based technology developed in some advanced agencies could be ready to normal breeders, right? Yeah. This is, of course, a quite complex issue. We cannot expect every breeder in the world, even in developed countries, will use all the technology. As what I said in my suggestion is that as a breeder, I'm a breeder. If I So we must be, as a breeder, if you want to use, you must be prepared materials as well as some necessary facilities and knowledge of this. Of course, agencies like governments or universities also should in education of technology transfer system should make most of the community of operators informed to see. But we don't expect everyone will use this. If most of them use this, it's fine. Fine enough, right?
I am borrowing on my experience from crop improvement in 6 different countries, which is Central America, South America, US, and here. I've been working in India for the past 5 years. My experience is that most breeders have not embraced the whole technology, right? So there is that divide that maybe these molecular biologists are taking, our geneticists are taking the role of breeders. Some of that is happening. From experience, the few who have embraced it are doing very, very well, and they're asking now, where was this a few years ago? An example is one of the breeders in Brazil, where I work as well. He said it never worked. But now he's almost forced to retire. He's begging to stay, and they have granted him because some of the things that he couldn't do before, he's doing them faster now. So it's there. And for the last question somebody asked about extension, our experience is that it is a little bit lacking, and what we have done to get to the farmers, we're now employing Pedialyte, the companies here, and Ambuja Cement in Gujarat. So we have them an ASI because this is how we can reach no formalized national extension program that I have seen per se. And also, Dr. Singh said most of these seed issues is crop by crop. I am basing my experience on groundnut. So that's the basis of my experience, okay?
Yeah, I think this is working. Is this working all right? Yeah. This is working. Friends, let me say it has been a wonderful learning process. Just 3-4 points which probably we would like to take as a take-home message. Innovations, as mentioned, so very important. Innovations for transformation. Genomics is one of those, just one, but it has to be genomics surrounded with many other enabling mechanisms which will allow this to happen. So let innovation lead the transformation, but it needs a little more support from various ways to enhance yield, productivity, profitability, sustainability, resilience to climate change. All these 5, 6 combined together to be seen as a complex message to be conveyed. And to convey a complex message, as the ICRISAT philosophy, humanizing science and delivering science to people, and the questions were probably around this. And I will say that just I want to emphasize on this point, when Borlaug was on his deathbed, some extension agent went to his house, said, "Sir, I have this technology. You will be happy to know about it. This is doing so well and all." Borlaug could listen to it, and he said, "Take it to the farmer soonest possible. As soon as you can, if this is working, it is a miracle, take it to the farmer. He is the one who needs it the most." Therefore, a strong message to you that if you have a technology proven to make a difference, take it to the farmer as soon as possible, even if necessary, break the rules, because the farmer and farmer's family is hungry and they are waiting for you. So I think this message is very strong and clear, and I will stay with that. Whatever it takes to humanize science, policy changes required, we have to go by that. The third thing is that probably our communication system is not good enough. We seem to fail on that. We must be able to communicate our good science in good ways, good language, and therefore Susan is not here, Henry is also not here. If I have, say, novel discovery, if my discovery is novel in sense of social terms, I would not fail in publishing in its Nature. So, do I have a novel social message put in my way of communication? If I have done it, if I have a strong message for that poor farmer, and I can do it properly, communicate it, Nature Biotechnology will publish it, Cell Biotechnology, Plant Biotechnology, all will publish it, and people will receive it. So do we have this communication? I think it is missing. I will say very strong support is required. And to do that, the next thing is the collaboration is the ultimate way. We noticed in Africa how it has played a very significant role in making major changes. And in doing that, obviously the integrated approach and multidisciplinary approach along the value chain will be absolutely necessary. Nothing is free in this world. If you have to go around the corner, if you need a dime, the dime must be in your pocket. And unless that your pocket has that dime, you can't go around the corner. And therefore, the investment at the right place for the right cause is absolutely necessary. Invest in good science and the science which can be with human face. And for that purpose, you prioritize your outcomes of science, science for development. And if that is the way, then the investments of quality will make the difference. So quality investment is important. Finally, please take it that all that is said is maybe looking beautiful, but there are many hurdles in the way. It may be a regulatory hurdle, it may be a social hurdle, it may be a cultural hurdle, all kinds of things, information gap and so on and so forth. But if we have to really break these hurdles, probably all we have to keep it in mind that if we have a heart with science to help the people, we will find the way. And this is what is not happening, and this is where the implementation comes into play. All stakeholders come into play, and so I will say, please develop that human touch to the development, and that will take you to the place where you need to reach. So thank you very much for the wonderful opportunity we had, and thank you, panelists. I look forward to seeing you, interacting with you more and more. The whole audience needs, the people need you, and we thank you so much. Thank you, Raju, for the opportunity, and thanks a lot. Thank you, friends. Sorry for keeping you waiting for too long.
No. So I would just like to mention that I'm sure that we had really great session, and thanks to all the panel members to share their views, vision, and having very nice discussion. Thanks also to all the delegates. So with these words, please join me in thanking both co-chairs and all the panelists once again.